Welcome to the deep dive. Today, we're really getting into the nitty-gritty, unlocking some secrets about how our body's most basic units, our cells, actually work and talk to each other.
Yeah, and we're focusing on cell membranes and action potentials.
Right. And these topics, especially if you're a first-year medical student listening, they can seem, well, pretty overwhelming at first, just a flood of details.
Definitely.
So our mission here in this deep dive is really to break these big ideas down, make them super clear, give you some mental hooks, metaphors you can actually use.
The kind of thing you could maybe explain to a curious kid, right?
Exactly. So you really get it, and it sticks.
Yeah.
Building that solid foundation you'll need for, well, everything else in medicine.
Absolutely. Because what's fascinating, what's really fundamental here, is that these two areas, they're basically the language of physiology. Understanding membranes and action potentials. It's kind of like learning your ABCs before you try to write, a novel. It just unlocks pretty much every process you're going to study.
Okay, makes sense. Foundational.
Get these down, and suddenly how our bodies function starts to seem much less mysterious, more logical.
All right, let's do it. So piece number one, the cell membrane. The cell's like smart boundary. So imagine your cell is like this tiny, busy house. The cell membrane, that's its walls, but not just any walls. It's more like a smart wall or maybe a security fence.
Good analogy.
Yeah, because it doesn't just hold stuff in. It's active. It's really selective. It forms this barrier, sure, but it's got these special gates.
Right, controlling who gets in and out.
Totally. It keeps the inside environment, tidy. It gets messages from the outside world. It can even send out its own signals like a doorbell or maybe an alarm.
And importantly, different parts of that fence can do different things.
Ah, okay. So it's specialized in different areas.
Precisely, depending on what that part of the cell needs to do.
Cool. So if it's a smart wall, what's it made of? What are the bricks and mortar?
Okay, so the main building blocks are lipids, and primarily we're talking about phospholipids.
Phospholipids. Got it.
Now, these are really interesting molecules, almost like tiny two-faced characters. We call them amphipathic.
Amphipathic means they have two sides.
Yeah, exactly. They have a head part that loves water hydrophilic, and then these tails that hate water hydrophobic.
Yeah.
Think of them like little tadpoles with water-living heads and water-hating tails.
Okay, tadpoles. I can picture that.
So what happens when you throw these tadpoles in water? The tails, because they hate water, they automatically try to hide. They all clump together in the middle, away from the water.
Makes sense.
And the heads, they happily face outwards towards the water on both the inside and the outside of the cell. This creates a structure, spontaneously, called the lipid bilayer, like a sandwich or a bubble wall with the tails hidden inside.
Wow. Okay, so it just naturally forms that structure.
Pretty much, driven by those hydrophobic interactions. But it's not just phospholipids.
Ah, okay. What else is in this wall?
Well, you've got glycolipids. These are lipids with sugars attached. Think of them like maybe little address flags or sweet decorations on the fence. They help with cell recognition.
Like ID tags.
Sort of, yeah. And then there's cholesterol. A lot of it, actually. Makes up maybe 45% of the lipid in some membranes.
Cholesterol. I thought that was just bad.
Well, it gets a bad rap sometimes, but in the cell membrane, it's crucial. It's like the temperature regulator or maybe the mortar in that wall analogy.
Okay, mortar. How so?
It modulates fluidity. When it's warmer, like at body temperature, cholesterol actually makes the membrane a bit less fluid, less leaky, stops it from falling apart.
Right. Keeps it stable.
But when it gets colder, it does the opposite. It stops the tails from packing too tightly and freezing solid. It keeps the membrane flexible.
So it works both ways. Keeps it in that just right zone.
Exactly, that Goldilocks zone. It's essential for maintaining the right level of fluidity.
So this mortar, this cholesterol, makes it stable. But does that mean the membrane is rigid? Or is there still movement?
Oh, it's definitely not rigid. That's a key point. It's dynamic. Very fluid, actually.
Okay, so how does that work with the cholesterol stabilizing it?
Well, a lot of the fluidity comes from the fatty acid tails themselves, the ones on the phospholipids, especially the unsaturated ones.
Unsaturated, meaning they have double bonds.
Right, cis double bonds.
Mm-hmm.
And these create kinks, like little bends, in the tails.
Kinks.
Imagine trying to stack bent sticks versus straight sticks. The bent ones don't pack together neatly, right? There's more space, more wiggle room.
Ah, I see. So the kinks make it harder to pack tightly, creating fluidity.
Precisely. So you have the unsaturated fats adding fluidity and the cholesterol kind of modulating it, keeping it functional across different conditions. It's a balance.
Okay, so we have this fluid shifting sea of lipids, but you said that the membrane has specific jobs like gates and signals. Lipids don't do that, do they?
Good point. No, that's where the membrane proteins come in. These are the real workhorses, the functional widgets or the tools embedded in or stuck onto this lagged C.
Right, the specialized parts.
Yeah, they do all sorts of things. They act as transporters, the doors and gates, anchors mooring the cell, receptors receiving signals, enzymes carrying out reactions right there at the membrane. They even help cells stick together. Huge variety of functions.
Okay, wow. Lots of roles. Are there different types of these protein tools?
Broadly, yes, we talk about integral proteins. These guys are really embedded in the membrane deep down, interacting with those hydrophobic lipid tails.
Like built right into the wall?
Exactly. You can't easily pull them out without disrupting the whole structure. You usually need detergents to isolate them.
Okay. And the other type?
Peripheral proteins. These are more loosely attached, sitting on the surface, often bound by weaker bonds.
Like a sign tacked onto the wall.
That's a great way to put it. You can often wash them off with gentler treatments, like changing the salt concentration or pH, without wrecking the membrane itself.
Got it. Integral are in, peripheral are on.
Basically, yeah. And this whole picture, the moving lipids, the embedded and attached proteins, that leads us to the famous fluid mosaic model.
Yes, I've heard of that. Singer and Nippleson, right. 1972.
That's the one. The name captures it perfectly. It's fluid because the lipids and many of the proteins can move around laterally, like drifting in that sea.
Okay.
And it's mosaic because you have all these different proteins dotted throughout the lipid background, like tiles in a mosaic pattern.
A sea of lipids with protein icebergs floating in it.
That's a classic way to visualize it, yeah. Though it's important to remember, not all those icebergs are freely floating.
Oh, what starts them?
Well, some proteins might be clumped together in functional groups. Others might be anchored to structures outside the cell, or, very importantly, tethered to the internal scaffolding of the cell, the cytoskeleton.
The cytoskeleton, like the cell's internal framework.
Exactly. A great example is in red blood cells. Their membrane needs to be super flexible, but also strong, right? To squeeze through tiny capillaries.
Yeah.
So their membrane proteins are linked to an underlying network of cytoskeletal proteins like spectrin and anchorin. This network provides support and shape, but still allows flexibility. It's like internal bracing for the smart wall.
And I guess if that bracing is faulty, things go wrong.
Absolutely. That leads us directly to clinical relevance. There are genetic conditions, like hereditary spherocytosis or elliptocytosis, where defects in these cytoskeletal proteins like spectrin or anchorin make the red blood cell membrane fragile.
So the wall is weak.
Essentially, yeah.
Yeah.
The cells lose their proper shape, become more spherical or elliptical, and get easily destroyed, especially in the spleen. This leads to anemia.
Wow. So a tiny defect in a structural protein has huge consequences.
It really underscores how crucial every component is. Structure and function are totally linked. And this careful structure, the smart wall, its main job is controlling passage, right? Transport.
Okay, yeah. Getting things in and out.
Exactly. Which connects to the bigger picture. Controlling that traffic is vital for cell survival for everything it does.
So how selective is this barrier? What gets through easily? What needs help?
Well, the lipid bilayer itself is a pretty good barrier against charged things like ions, sodium, potassium, calcium, and also large polar molecules like glucose. They just can't get through on their own.
Too big or wrong charge?
Basically, yeah.
Ah.
They don't mix well with that hydrophobic core. But small, uncharged polar molecules like water itself or urea, they can sneak through to some extent. And small hydrophobic molecules, things like oxygen, carbon dioxide, they slip through easily.
Like dissolves, like kind of.
Sort of, yeah. The hydrophobic ones pass through the hydrophobic core readily. But for most things the cell needs, or needs to get rid of, it requires help. Proteins again.
Okay, the transporters we mentioned earlier.
Right. And broadly, transport happens in two main ways, passive and active.
Passive means no energy needed.
Correct. It's movement down a gradient, like rolling downhill. Either a concentration gradient from high concentration to low, or an electrical gradient towards the opposite charge.
So simple diffusion, like O2 moving, that's passive.
Yep. And then there's facilitated diffusion. Still passive, still moving downhill, but it needs a protein helper because the molecule can't cross the lipid bilayer alone.
Okay, and those helpers are?
Two main types. Carrier proteins, which are like those revolving doors. They bind the molecule, change shape, and release it on the other side.
Like the band 3 protein in red blood cells you mentioned.
Exactly, swapping chloride and bicarbonate. That's facilitated diffusion via carrier. The other type is ion channels.
The tunnels or gates.
Right. They form a pore through the membrane. When open, specific ions can just flow through really quickly, still down their gradient. And as you said, they're often gated.
Gated by
Yeah.
chemicals or voltage?
Yep. Ligand-gated channels open when a chemical key binds. Voltage-gated channels open or close when the membrane's electrical charge changes. These voltage-gated ones are super important for, well, what we're getting to next.
Action potentials. But first, what about going uphill, against the gradient?
Ah, yes, that requires energy. That's active transport.
Pushing the boulder uphill?
Good analogy. It needs energy, usually from breaking down ATP, either directly or indirectly.
So primary active transport uses ATP directly.
Correct. And the absolute classic, the superstar example here, is the sodium-potassium pump, the Na plus MASK-K plus ATPase.
The pump everyone learns about.
And for good reason. It's this tireless little machine in almost all animal cell membranes. For every ATP molecule it burns, it pumps 3 sodium ions out of the cell and 2 potassium ions in.
3 Na plus out, 2 K plus in. Okay, why is that so important?
Oh, massively important. It maintains those steep concentration gradients, low sodium inside, high potassium inside. These gradients are crucial for cell volume, for pH balance, but maybe most importantly, they store potential energy, energy that's used for electrical signaling and for...
Secondary active transport.
Exactly. But first, just think about the energy cost of the Na plus MASK-K plus pump itself. In nerve cells, it can burn through like 25%, maybe even 70% of the cell's total ATP.
Wow, 70% just for this one pump.
It's a huge energy investment. Shows how vital maintaining those gradients is. It also means things that disrupt ATP production, like lack of oxygen during a stroke. Well, this pump fails, gradients collapse, and cells like neurons die quickly.
That really puts it in perspective. Okay, so it builds up this big sodium gradient outside the cell.
Right, like pumping water uphill into a reservoir.
Mm-hmm.
And secondary active transport taps into that stored energy. It doesn't burn ATP itself.
It uses the flow of sodium back downhill.
Precisely. It couples the downhill movement of sodium into the cell with the uphill movement of something else, like that water flowing downhill, turning a wheel to lift something else up.
Clever.
Yeah. If both molecules move in the same direction, it's symport, like the NAM plus glucose cotransporter in your gut lining. Sodium flowing in pulls glucose in with it, even against glucose's gradient.
So sodium drags glucose along for the ride.
Pretty much. If they move in opposite directions, it's antiport, like the NAM plus NaNO2 plus exchanger, which uses sodium flowing in to push calcium out, or the NAM plus H plus exchanger getting rid of acid from the cell.
Okay, symport together, antiport opposite. Got it.
And just quickly, a uniporter is just a transporter, often facilitated diffusion, that moves only one type of molecule.
Right, so we've got the membrane structure, the lipids, the proteins, the ways things get across passively and actively.
We've set the stage, yeah. Controlling the internal environment, building up these ion gradients.
But this raises the big question. How does all this careful ion shuffling lead to something as fast and dynamic as, like, a nerve impulse?
Exactly. How does this traffic control become electrical communication?
Right. Which brings us neatly to part 3, action potentials, the cell's electrical language. So all this ion movement we talked about, especially the pump keeping sodium out and potassium in, creates an electrical difference across the membrane, right?
Absolutely. Every cell has it. It's the resting membrane potential. The inside of the cell is negative compared to the outside. For a typical neuron, maybe around negative 70 millivolts.
Minus 70 millivolts.
Tiny.
Okay, and that's like a charged battery.
That's a perfect way to think about it. Or a coiled spring. It's stored potential energy, ready to be released. It's mostly due to potassium ions. There's lots of K plus inside. And the membrane at rest is quite permeable to K plus through leak channels. So K plus tends to leak out down its concentration gradient, leaving negative charges behind inside.
And the Na plus K plus pump keeps putting the K plus back in and the Na plus out to maintain this whole setup.
Constantly working in the background, yes. It maintains the gradients that fuel the battery.
So the signal happens when this resting potential changes.
Exactly. The signals are the changes. If the inside becomes less negative, say, goes from negative 70 millV towards 0, maybe to negative 50 millV, that's depolarisation, the battery charge is decreasing.
Usually caused by positive ions coming in, like sodium.
Typically sodium or calcium entry, yes. If the inside becomes more negative, say, negative 70 millV down to negative 90 millV, that's hyperpolarisation, the battery is sort of overcharged.
And that's usually potassium leaving or chloride entering.
Correct. Opening K plus or Cl channels often causes hyperpolarisation.
And controlling these changes comes down to those gated channels again.
Precisely. Especially the voltage-gated ones for action potentials. Remember, they open or close based on the membrane voltage itself.
Okay, so let's put it together. The action potential, the nerve impulse.
Right. Think of it like a very fast electrical wave, or maybe dominoes falling along the nerve cell's membrane, specifically down its axon.
Dominoes. I like that. And it's all or nothing.
Absolutely. Once you tip that first domino past a certain point, the threshold, the whole line goes down. No halfway.
So how does it start? How do you tip the first domino?
It starts with signals coming in from other cells, usually as synapses. These signals cause small, local changes in potential, often small depolarizations. These spread to a region called the axon hillock.
The trigger zone?
Kind of, yeah. If enough of these small depolarizations arrive around the same time and sum up to push the membrane potential at the hillock up to the threshold, typically around meg 55 milliviter,
Then boom.
The all-or-nothing action potential is triggered. Voltage-gated sodium channels fly open right there.
Okay, so what are the phases? What happens after threshold?
First phase, depolarization, or the upstroke. Those voltage-gated Na+ channels open incredibly fast. Sodium ions rush in, driven by both concentration and electrical gradients. This makes the inside rapidly shoot up, becoming positive, maybe plus 30 millivolts.
The first domino falls hard.
Exactly. But then, very quickly, those same Na+ channels slam shut and inactivate. They can't reopen immediately.
Okay, sodium influx stops. Then what?
Phase 2. Repolarization. Around the peak, voltage-gated K+ channels, which open more slowly in response to the depolarization, are now fully open. Potassium ions rush out of the cell, down their gradients.
Positive charge leaving.
Right. This outflow of positive charge brings the membrane potential rapidly back down towards negative values. The cleanup crew, restoring order.
Back towards resting potential?
Even past it, briefly.
Yeah.
Phase 3 is often hyperpolarization or the undershoot. Those K+ channels are a bit slow to close, so for a moment K+ permeability is even higher than at rest, and the membrane potential dips more negative than negative 70 milliV.
Okay. And then it settles back to rest.
Yes, as the voltage-gated K+ channels finally close and the resting permeability and the No plus K plus pump take over again. But that brief undershoot period is important.
Why is that?
It contributes to the refractory period. During the action potential itself, absolute refractory period, when Na+ channels are inactive, and this undershoot, relative refractory period, the membrane is difficult or impossible to stimulate again immediately.
Like the dominoes need to be reset.
Good analogy. That ensures the signal travels one way down the axon and limits the firing frequency.
Okay, so that's one spot on the membrane firing. How does it travel? The domino effect.
Pretty much. That strong depolarization during the upstroke at one point electrically affects the patch of membrane right next to it. It pushes that patch to threshold, causing its voltage-gated Na+ channels to open.
Ah, so the wave propagates itself.
Exactly. The depolarization jumps from point to point along the axon, regenerating itself without losing strength. Like lighting fuses down a line.
Cool. And what happens when this wave reaches the very end of the nerve, the synapse?
When the action potential arrives at the axon terminal, the depolarization there opens voltage-gated calcium channels. Calcium ions, Ca2+, rush into the terminal.
Okay, calcium's the key here.
Yes, this calcium influx is the trigger that causes vesicles, little sacs filled with neurotransmitters, to fuse with the membrane and release their contents into the synaptic gap.
The chemical messengers jump the gap.
Right. The electrical signal gets converted into a chemical signal to communicate with the next cell. That neurotransmitter then binds to receptors on the next cell.
And potentially starts the process over again, causing a depolarization or hyperpolarization in that cell.
Exactly. If it causes depolarization, making the next cell more likely to fire, it's an excitatory synapse, an EPSP. Often involves opening Na+ channels.
The go signal.
Yep. If it causes hyperpolarization, making the next cell less likely to fire, it's an inhibitory synapse, an IPSP. Often involves opening K+ or Cl channels.
The stop signal.
Right, and most neurons are constantly receiving thousands of these go and stop signals, summing them all up to decide whether they will fire an action potential. It's complex integration.
Wow. From lipids and proteins forming a barrier to these incredibly fast, complex electrical signals controlling everything.
It really is amazing, isn't it? Just the
Yeah.
sheer elegance of how this relatively simple structure, the lipid bilayer, decorated with these specific protein machines, can orchestrate everything from just keeping a cell intact to, well, generating the thoughts you're having right now.
Yeah. Form and function perfectly matched.
Totally intertwined, building incredible complexity from these fundamental molecular interactions.
So bringing it back to you, our listener navigating medical school, what's the big takeaway? Why does mastering this matter so much?
Because these concepts, membrane transport, resting potentials,
Yeah.
action potentials. They're literally everywhere in medicine. Nerve conduction in neurology, muscle contraction in cardiology, and, well, everywhere. Hormone signaling, immune cell activation, how drugs work at a cellular level. It all comes back to this.
So getting these fundamentals down, using these kinds of mental models and anchors.
It's not just about passing the next exam. It genuinely provides a framework for understanding health and disease throughout your entire career. It makes sense of the why.
Right. Okay, so here's a final thought to leave you with. Just pause and consider how many things you do without thinking. Blinking, breathing, your heart beating, even just feeling the chair you're sitting on. All of that, every sensation, every movement, every heartbeat is basically just a beautifully choreographed sequence of these tiny electrical action potentials firing along nerves and triggering responses, all thanks to the smart walls of your cells. Isn't it kind of mind-blowing that something happening at such a tiny molecular scale generates everything we experience and do?
It really is profound when you think about it that way.
So as you continue learning, maybe keep an eye out. What other complex systems in the body, maybe ones that seem totally unrelated at first glance, actually depend fundamentally on these same principles of membrane barriers and electrical signaling? Where else does this alphabet show up?